You've seen the headlines, human embryos from skin cells for the first time ever. This is a huge breakthrough in science, research, and the future of fertility options for a lot of patients. But what does it actually mean? Researchers at the OHSU Center for Embryonic Cell and Gene Therapy have created human embryos from skin cells. Again, this is a first time ever breakthrough in science. Here to explain the research is one of the lead authors, Dr. Paula Amato. She's going to help us understand the study and understand what it means for fertility and the future. Welcome to the Brave and Curious podcast. I'm your host, Dr. Lora Shahine, a double board certified OBGYN and reproductive endocrinologist helping people build families for over 20 years. And today I am curious about this incredible breakthrough, the development of human embryos from skin cells. I am so excited to have our guest, Dr. Paula Amato, professor of obstetrics and gynecology in the School of Medicine and division head of the Department of Reproductive Endocrinology and Infertility at OHSU in Portland, Oregon. Originally from Toronto, Canada, she did her medical degree and her OBGYN residency at the University of Toronto. She completed her fellowship in reproductive endocrinology and infertility at the University of California in San Diego. During her tenure at OHSU, Dr. Amato has served as the president of the American Society of Reproductive Medicine and focused her research on metabolic endocrine interactions, environmental impacts of reproductive health, and stem cells. Her work in the OHSU Center for Embryonic Cell and Gene Therapy with a team of scientists and researchers have led to this published work and is quite honestly shaking up the fertility community. So Dr. Amato, thank you so much for joining us today. I'm really excited to talk about this.
Thank you for having me. I'm really looking forward to it.
Wow, I saw all the headlines. So this is like the middle of October. This study came out the very last week of September, and it was just all over the place. You know, human embryos made from skin cells. And I started reading it, and I found the paper, and then I said, wait a minute, I know Paula. This happened just down the street. You know, I'm in Seattle. I'm like, this happened down in Portland. And so thank you so much for taking your time to come and talk to me about it, because listeners are really curious. You know, my patients are asking me, what does this mean for my fertility? So at first, you know, please just tell us what the study is and, you know, what you guys published on.
Sure. Well, first I have to say, as usual, the headlines, of course, are always a little more sensational, right, than the actual study. But it is quite complicated, the science part. So I'll try to and explain it best I can. But the idea is, as we all know, as women get older, their egg number and egg quality decreases until eventually they run out of eggs. And one of the main causes of infertility is advanced maternal age. So for women who are older, or for women, for example, who've undergone previous cancer treatment with chemotherapy, they may have run out of eggs and no longer have the opportunity to have a child that's genetically related to them. So the idea with this study is, what if we could take the skin cells from one of these women and, you know, remove the nucleus, which contains the DNA, and transfer it into a donor egg from a young donor that's had its nucleus removed? Could we then induce that egg to, one, get rid of half the chromosomes, because of course the skin cell has 46 chromosomes, and a mature egg has only 23 chromosomes, which then combines with the 23 from the sperm to create an embryo with a total number of 46 chromosomes. So that was the first challenge.
Let me pause for a second, because I really want people to understand, and I want to think about, I actually had to like read the study and kind of go back to my like biology class. So really basic, you know, our genetic code is on chromosomes, and those are those little X's in our biology class.
Yeah.
And it's like stacks of letters. That's like our genetic code. And
right.
The only way that embryos are created is an egg cell and a sperm cell, and they carry the genetic code. And what's different about these two cells that's different from all the other cells in the body is that the egg and sperm cell can get down to half of the genetic code, like half of the copies of the chromosomes. And then when the egg and sperm cell come together, they each give half of their DNA on, like, that's the 23 pairs of chromosomes in all of our cells.
Absolutely correct.
So skin cells can't do that. They're carrying around these 2 copies, right? And I actually, like Paula, I literally was like drawing chromosomes on a piece of paper, like going through this just to remember, because I really want to understand. And so when women and men, when you don't have sperm and egg cells that are working anymore, you don't have the ability to split chromosomes and reproduce.
That's right.
And if you could somehow get the genetic code from other cells and have them work in cells like eggs and sperm, then maybe this person's genetic code could be passed on. That's like kind of the basics, right?
Absolutely correct.
And then the other thing that I really helped me, and I drew it out, I had like colored markers and everything, is that the chromosomes and the DNA are in this little ball called the nucleus in our cells.
Right.
So that's where the bulk of the genetic code is. And so what you did is you took the genetic code from a skin cell in the form of the nucleus. You took this little ball out of the skin cell. You took the nucleus out of an egg cell.
Right.
Right?
Right.
And then you took the skin cell DNA, the nucleus, and you put it into an egg cell, because the egg has the ability to split the chromosomes in order to fertilize with sperm.
That's right.
So it's like you are, because right now the only way that people can have a baby if they don't have any eggs left is use donor egg.
Right.
Right. Or, of course, adoption or other things. Of course, there's many ways to build a family, but a lot of my patients are, using donor eggs, but that is the donor's DNA, right? The donor's nucleus.
Right.
And so if you can take an older woman's DNA from her skin cells and put it into an egg cell and allow. So again, I hope that that's helpful for listeners. And I just have to kind of break it down, because even in some of the articles, it's like a little bit confusing, but you got to get the big picture, how egg cells and skin cells are different. And then what is so amazing is that this is the first time this has ever been done in humans.
That's right. Yes, you did absolutely great job of explaining it and trying to simplify it. It is very complicated, even for me, as we've already discussed. But that's the idea. Yeah. And donor eggs, of course, is a great way to build a family. So nothing against family building through donor eggs. But there is quite a strong desire for people to have a genetically related child. So that's where this kind of work comes from. That's the impetus for it. Yeah.
And this has been done in animals, right? Like this is what was done with Dolly the sheep in 1996. Like I remember those big headlines.
Yes.
For people that weren't alive or reading news back then, like we were, Paula, like this sheep named Dolly was like cloned. So it was an adult sheep, skin cells, and then reproduced. Was it similar to this?
Very similar. So the part about transferring the nucleus into a donor egg cell is called somatic cell nuclear transfer. And that part of the procedure is exactly the same. Now, of course, Dolly, as you said, is a clone. So Dolly was genetically identical to her mother, if you will. This is a little bit different in that we're trying to get rid of half the chromosomes, so then what's left can recombine with sperm and create an embryo that's genetically unique and has a combination of chromosomes from both the sperm and from the person providing the skin cell.
Okay. And this has been done with sperm cells in the past, no? For a skin cell to a sperm cell?
Yes, that turns out to be technically more difficult. So that hasn't quite been achieved yet. For a variety of reasons. Yeah, it has been done, as you said earlier, in mice. So there's kind of two different approaches. One is the approach that I just described, and it's called somatic cell nuclear transfer. But there's also a group in Japan and several other groups working on a technology called induced pluripotent stem cell reprogramming.
Oh dear.
Very long name.
Yes.
But it's basically taking the skin cell like we did, but trying to reprogram that skin cell to become a stem cell. And then, of course, stem cells can become any kind of cell in the body. So then programming it to become an egg cell. Wow. And it turns out that works in mice. And our technique works in mice as well. But neither of those two approaches have been successful yet in humans.
Well, and I feel like, I mean, I watch movies too, you know, like Gattaca and like Mission Impossible and things. And it just sort of seems like people are doing genetic stuff, you know, all the time. And that, you know, this is like so easy. But how long have people been working on this? And even just at your program at OHSU, you know, what is sort of the time that's been put into this and like the, you know, frustrations and, you know, what did it take to kind of get here?
Yeah. So me personally, I've been at OHSU for 18 years. And the entire time I've been here, I've been collaborating with my main collaborator, Dr. Shukrat Matolipov. He's senior author on the paper. And we've been working on these sort of innovative assisted reproductive technologies to treat primarily age-related infertility, but also to prevent transmission of genetic diseases. So over a decade ago, we successfully did the somatic cell nuclear transfer in human eggs and embryos for the first time. And this particular project, project is called in vitro gametogenesis, or IVG. That's trying to make egg cells from skin cells. We've been working on this project for about 5 years.
Wow. What was it like? Yeah. Who made the discovery and like called everybody on the team? Like, what was that like?
Well, you know, as most research teams do, we meet regularly. We meet every week. And so, and this is what we do every day, like somatic cell nuclear transfer and that stem cells, you know, this is our bread and butter. And I
think
this
is so amazing. You're just like, oh yeah, we just do this every day. This is so amazing. Okay, cool. Yeah.
And I think it was Dr. Matolipov who said, hey, you know, what if we combine, you know, somatic cell nuclear transfer, but like we do put a little twist on it and try and activate that egg to get rid of half the chromosomes? Could we then, you know, fertilize that egg and develop a unique embryo that has a combination of chromosomes from the person providing the skin cells and the person providing the sperm? And you know, like most of our things, they start with an idea, then we try it initially kind of in a mouse model and an animal model. And then if it works in mice, then we move on to human eggs and human embryos because, you know, obviously it's not easy to get human eggs, as you know, and we're eternally grateful to our egg donors who participate in the research. But we don't, you know, we're really careful not to use too many eggs or produce too many embryos unless we're pretty sure we're going to get some really valuable information from those experiments that we can then use to treat patients clinically.
That is really important. How are you able to do this work ethically? You know, what does it take to have oversight and to kind of work really closely to make sure you're doing this in a very respectful way in order to help people?
Right. Yeah, very good question. And, of course, we can't do this type of research, or you can't do this type of research in all parts of the country, Eden. But we're very lucky in Oregon that the regulatory environment is such that it allows us to do this research. And, pretty much we have to go through, human subjects approval. So there's an institutional review board that reviews all the research at our institution, in my case, Oregon Health and Science University. And we have to propose the project and they have to review it and they have to approve it. And, you know, that particular review process took quite a long time, as you can imagine.
Yeah.
And then, you know, they'd let us recruit egg donors specifically for research. So, of course, all fertility clinics or most fertility clinics these days recruit egg donors for fertility purposes. So this is exactly the same process for the donors as that, except what we do with the eggs is a little bit different. In this case, we're not transferring those embryos to establish a pregnancy, but we're trying to use that information to ultimately help treat patients with infertility.
Yeah. And you're really transparent with the women that are donating their eggs. And then I would assume, of course, the men that are donating their sperm to. And so it's just really important to realize that this is completely done under, you know, guidelines and an institutional review board, and the transparency, I think, is really, really important.
Absolutely.
We're going to get back to learning from Dr. Amato in just a second, but I want to thank you for being here. Be sure and follow the show wherever you're listening. If you want to watch the interview, you can see it on my YouTube channel, Dr. Lora Shahine. And take a moment to give us a 5-star review and comment with something that you find valuable. It really helps others find the show, and it means so much to us. Now, let's get back to learning. How is this different from three-party parenting, or what's it called? There's the headlines have been out there like 3-parent IVF, I think is what people call it. I can see people being confused. And how is that different from what you're doing?
Yeah, that's another one of our research projects. But you're right, it is kind of similar but different. So, and 3-parent baby, of course, or IVF is a little bit of a misnomer, of course, because usually there's only 2 parents.
Yes.
But in that case, what we're doing, so this is for women who still have some eggs, right? But maybe they have a mutation in their mitochondrial DNA. So another sort of point of education for your listeners is all our genetic material is in the nucleus, like you said, and most of the genes that code for most of our traits those genes are located in the nucleus, except for the mitochondrial DNA. And that is outside the nucleus. And that is very unique in that we inherit it exclusively from our mothers. Okay. Mm-hmm. Mm-hmm. So, but that DNA, it just provides energy to our cells. It doesn't really code for any traits. So, but there are some women who carry mutations in their mitochondrial DNA, which can lead to very serious diseases in the children. So the idea with mitochondrial replacement therapy, we call it, is to take an egg from one of those women, again, remove the nucleus from her egg, and then transfer it into a donor egg that's had its nucleus removed, but has healthy mitochondrial DNA left over in that egg cell, and then fertilize that now reconstituted egg with sperm from the patient's partner to create an embryo, and then transfer that embryo into the patient's uterus to establish a pregnancy and hopefully deliver a healthy baby that doesn't have mitochondrial disease.
Yeah.
Now that research is much further along. So there have been babies born already from that particular technique. And again, the main difference between that and what we did with the skin cell is the egg cell already has only 23 chromosomes. So we didn't have to get rid of half of them, right? We just took 23, put them in the donor egg that's had its own 23 chromosomes removed, and then fertilized the egg. So it's a little bit simpler, actually.
Yeah. And to just clarify, and you absolutely just explained it really well, is that, remember, egg cells are used to doing this. Like egg cells are 23, half of the genetic content. They're going to fertilize with sperm, which is half of the genetic content. That's great. What's been done with 3-party IVF, or however you want to say it, is it's just as a way to not inherit these very rare diseases from the mitochondria, which is out in this little DNA suitcase in the cell. I'm like, what could I say? Just outside of the nucleus, and totally different. But you could see this is how confusing this could be to people.
Right.
And another really important thing that you said is there have been babies born with this way to decrease passing on mitochondrial diseases. That we're not doing that in the United States, though. Is that correct? It's only in the UK.
That's correct.
And other countries.
Yeah.
But what you have done is absolutely not ready for prime time. So explain how far you guys got in the lab, so people are clear that this isn't something that they can go and ask their IVF clinic for right now.
Right. So with respect to the IVG project, which is like trying to make egg cells from skin cells, it's very, very preliminary. And it might be years, maybe even more than a decade before it ever gets to the clinic. So the problem is, even though we made embryos that had genetic material from the sperm and from the person who donated the skin cell, those embryos were all chromosomally abnormal. So they had the wrong number of chromosomes, or in some cases, the wrong combination of chromosomes. So none of those embryos could be transferred into a uterus to produce a healthy baby. So now we have to figure out, how do these embryos, separate and divide, or not embryos, but chromosomes separate and divide? And how do we make sure that one chromosome from each pair of chromosomes is what's shed? So one chromosome number 1 and one chromosome number 2 and one chromosome number 3, etc. Not some random combination of 23 chromosomes, because obviously that's not going to work.
Yeah. So, I mean, but it's an absolute breakthrough in the fact that you had an egg and a sperm in the lab, and that egg had DNA nucleus from a skin cell, right?
Yep.
And then you fertilized them together.
Yep.
I would assume that you did ICSI for all my IVF patients out there, the
Yes.
intracytoplasmic sperm injection to help.
Yes.
I don't know. I just, nice to, because people are curious.
Yep.
And then when the egg and sperm are together, not all of them started to duplicate, right? Because the cells have to divide and continue to grow.
Right.
But then you can sometimes get the, egg and sperm, the cells are dividing, they're becoming blastocysts,
Right.
which are these mature embryos that are, 100 to 200 cells. And at that point, you were able to biopsy a few cells from these embryos, just like we do in our IVF lab every day. And then you froze the embryos, I guess, and got the chromosome report back. And I believe in the paper, it was 9 embryos out of hundreds of cells. I mean, this is already an inefficient process, but this is really preliminary.
Yeah.
I just want everyone to know.
Yeah, it was very inefficient. You're right. So not all the eggs fertilized, not all the embryos made it to blastocyst. Only 9% of the embryos made it to that stage.
9%.
And of the ones that made it to blastocyst, all of them were abnormal, chromosomally abnormal. Okay. So it's an incremental step, I would say. And I think we'll get there eventually, but we still got to figure out a lot of things.
Yes, it's still amazing, but again, not ready for prime time.
Right.
But for we think ahead. What does this mean for the future of fertility? Who could benefit from this potentially in the future?
Right. So we already mentioned older women, or women who've had chemotherapy or were born without eggs. So for that group of patients, this would be applicable. The other interesting group would be same-sex couples. So right now, as your listeners probably know, if you have a female same-sex couple, usually they have to use donor sperm. If you have a male same-sex couple, they have to use an egg donor as well as a gestational carrier. So this would allow potentially for same-sex couples to have a child that's genetically related to both partners. So if you could take the DNA from the skin cell of one of the male partners, say, and transfer that DNA into an egg, donor egg, and then fertilize that egg with the other partner's sperm and make an embryo, now that embryo would have DNA from both male partners.
Yeah. I can just see the incredible change, especially for, just with how many people really are struggling with premature ovarian failure, maybe diminished ovarian reserve, even at a young age. I mean, I feel like there's this sort of feeling like, oh my gosh, it's women's fault for waiting too long to get pregnant, and we're all waiting until later to get pregnant. And so, this is another option. It's kind of like what egg freezing was maybe a decade or more ago. And now you don't even have to go through that process. You just kind of give a skin cell and are able to reproduce. I mean, it really, I'm not saying it very clearly because my mind is working so quickly and just thinking about how many people this really could benefit.
Yeah.
That would be and then I was thinking to myself, well, it's the end of egg donation, but it's actually not, because you do need an egg.
Yes.
It's just that you're passing on different nuclear DNA.
Right.
So I thought that was pretty interesting too.
Yeah, that is one of the limitations of our approach. It still requires somebody to donate eggs. So whereas if you could reprogram the skin cell without having to use a donor egg, obviously that would be beneficial. In fact, that would change IVF completely, right? We would not have to do ovarian stimulation egg retrieval anymore. You could just make eggs from your skin biopsy, which is kind of crazy when you think about it. But yeah, getting back to your egg freezing point, it is true. If more people had access to egg freezing, then, you know, we might not need to do IVG. But of course, not everybody has access to it, or, you know, there are people who don't have the opportunity for whatever reason or are born without eggs. So I think there would still be a role for this IVG approach.
What about ethically challenging topics that have come up, like even when we were doing the review, kind of talking to your colleagues, I could see this making a lot of people very uncomfortable. You know, could this be that someone, you know, takes my hairbrush and like all of a sudden I have genetic kids that are running around? I mean, I can certainly see the sci-fi movies coming from this, but I mean, I could really see people having concerns. What have you guys talked about?
Yeah, a lot of different issues, including that, of course, sort of inadvertent parenting when you might not even know about it. I mean, that's quite far-fetched probably, but not impossible to imagine. Also, like, who would have access to technology? It's likely not going to be cheap when it becomes available. And just like IVF now, there are inequities in terms of who can access that technology. Another issue would be, you know, even though someone who's in their 60s now can use this technology, for example, to reproduce, somebody still has to carry that pregnancy. So if now we're encouraging women to become pregnant in their 50s and 60s, I'm not sure that's a great idea either, of course, because as we both know, pregnancy can be very complicated in women who are older and riskier, both for the moms and for the babies. So, and of course, not everybody can use a gestational carrier, and that's, you know, controversial enough in and of itself. So, you know, these are some of the things we have to keep in mind. But I think like most of these technologies and other technologies, like just like, you know, military technology or nuclear technology, they're developed often for very benign purposes or for good purposes, but they could always be misused. And we have to be definitely aware of that possibility and be talking about all the ethical issues. But I guess I'm of the opinion that we shouldn't stop the progress. And even if we wanted to, honestly, we probably couldn't. You know, we have to consider all these issues, but I think the technology needs to be moved forward because I do think there is a role for it in helping patients have genetically related children.
Yeah, I do. I hear you. I do feel in our field that sometimes technology develops faster than what we really know what to do with it. And a lot of ethical challenges come up, of course. And there's been a big call in society to have more oversight over IVF clinics and reporting, you know, mistakes or reporting just all sorts of outcomes. And there's a real worry from a lot of people in society that it's just the wild, wild west and that there's not a lot of oversight. And I'm curious, as you're being a past president of our American Society of Reproductive Medicine, being involved in this really cutting edge technology that has so much hope for the future, but yet, of course, could be used in a different way. Where do we come together where we don't hinder science and development and research, but also try to do it ethically? How do you do that?
Very challenging, but I think the
just real quick answer, Paula. You know, I think
the field actually has done, and I'm sure people might disagree with me, but I think we've done actually a pretty good job to self-regulate. It's a largely self-regulated industry or field, whatever you want to call it. And for the most part, I think the people practicing are quite ethical, and we consider, you know, we have an ethics committee as part of the American Society for Reproductive Medicine. Many clinics have, you know, their own independent ethic committees. And these things come up and we talk about them. Mm-hmm. I think what you said earlier, the transparency is important. But I think really we're the best people to regulate ourselves because we understand all the nuances. The last thing I think I want is the government kind of trying to tell us what to do, especially because they don't understand, what we do and the technology very well. So I would say I think we've done a pretty good job. Are we perfect? No. And anytime you're dealing with humans, mistakes are going to happen. But I think it's important, like you said, that we admit those mistakes when they happen. And the important thing is, understanding why they happen, trying to prevent them from happening again.
Yeah, thank you for that. It's exciting, but it's also trying times. And you're right. I mean, there are politicians and lawyers that are making decisions on the reproductive health of women that have never counseled women like you and I have in certain situations. And it's hard because I do try to believe that everybody's coming at it, trying to help and sort of be ethical. But I think you have to have a lot of knowledge to even make decisions like that. And you've got to have people like you in the room that understand the science to contribute to that and do it. Something that is just so exciting, but also could be seen as ethically challenging, right?
Sure.
So it's a real balance.
Yeah. And people have like varying religious beliefs, for example, or moral beliefs, and those come into play as well. And, I think we should respect everybody's opinion. I guess where I draw the line is like, trying to impose those beliefs or opinions on other people. That I think, is not appropriate in this country or anywhere, really.
Yeah, I hear you. All right. So Paula, what is next? What are you guys working on or clarifying in the, repeating or, what's next in the research for you guys?
Oh my God, so many exciting things. So of course, we're continuing the research on the mitochondrial replacement therapy. We're definitely going to continue the research on in vitro gametogenesis or IVG. And like I said, we have to figure out how to make those chromosomes kind of separate faithfully so that we get embryos with the correct number of chromosomes and the right chromosomes. And then we have other projects going on, such as gene editing in embryos to try to prevent diseases being transmitted from the parents to the child. So all very exciting. I feel really sort of privileged to work in this field and to help patients and to practice in a state like Oregon and at OHSU. And in combination with Spring Fertility, which is the IVF clinic that we work with, to be able to do this research, which I hope will help people.
Yeah, absolutely. Absolutely. So Paula, this is incredible. I'm so excited to talk to you, one of the researchers, and the headlines say we have made human embryos from skin cells.
Yeah.
So what did the headlines get right and what did the headlines get wrong?
So the headlines were a little bit sensational. So this technique, it is true that if it, is successful, then it would revolutionize the way we practice IVF and infertility. However, it's not quite ready for prime time yet. I think we've made a small step into showing that this might be possible in the future, but I think we're many years away before it can be applied to patients in the clinic.
Well, thank you so much for being here today and answering all of my questions. Just incredible what you're doing. Thank you, Paula.
Thanks for inviting me. My pleasure.
I hope you enjoyed this episode. It is incredible to talk to one of the scientists that just had a huge breakthrough in research and the future of genetics and fertility. Remember, this is not ready for prime time, but this could be a way to help people pass on genetic material in the future in a way that is not available today. Share this episode with a friend that you know is curious about all of those headlines. It says human embryos from skin cells, and have a discussion about it. This is such an interesting time. It's incredible. But remember, it is not available in your IVF clinics today. This is just science so far and research, but it also brings up really incredible ethical discussions. Thank you so much for being here. Be sure and follow the show wherever you're listening. Comment with questions that you have. Give us a review. Share with a friend. This is how people find the show. Thank you so much to my team at Audiotocracy for producing another wonderful episode. And until next week, stay brave and curious.